Let me see.
Oh, is there like a deeper?
No. Mm-mm. Okay, let's share screen. Not what I meant to do. I'm just realizing that I need to get on the Kahoot thing, and maybe I can do it through the app. This still takes less time than how I was doing it, so at least there's a OSU. Why is it doing this? I've already have an account. What was that? Yeah, what the heck is this? This is what I don't even know what's going on anymore. This isn't the right thing. Hopefully. Okay. All right. So does anybody have any questions over, projects or content or anything like that? No? Okay. Well, then you get to sit here and watch me work on this some more. I really thought that would fix this. Maybe I just need to close it out. No, it's really not working. So weird. I don't understand that. It's... Oh my God, how many times are you going to ask me? Okay. Why is it not working? So whatever, I guess I'm gonna have to do it the old school way because I can't connect. It's probably because the internet access sucks so bad down here. I can sometimes not connect. Here we go. That's a problem too. What you guys will find out, hopefully it won't be bad. Some semesters, I don't know what it is. When we're down here taking our exams, some people's computers just won't connect. And there's no testing center at Lake. So you get to take the paper test if that happens. So I have to sometimes bring like 10 paper tests. And it is what it is. I don't have any control over that, unfortunately. Don't want this. Okay, so chapter 2. Okay, so we're gonna try this. It won't have like the music, fortunately, but this at least I can like write stuff on here instead of like on the board, and it'll be up here instead, because I don't have much options with that. But no music, I can play it through my iPad. But that's it. Whoa. Hopefully that'll go a little smoother next time, so don't waste as much time. Okay, let's go and get started. All right, which type of chemical bonds involve sharing electrons? All right, so that's covalent. That's exactly right. And remember, the way that's best for me to remember that like you're cooperating, you're working together, so you're sharing. So good job, guys. All right, hydrogen bonds now. Okay, so that was weird. I usually expect a different sound to happen. So the answer is hold water molecules to one another. Why isn't it the blue one? Because this is the popular answer, is the blue one. Okay. It's not the blue one because the blue one says form between hydrogen atoms in different molecules. When we have hydrogen bonds, it's not between two hydrogens, because that would be positive to positive. And so it would have to be a hydrogen to something negative, right? Because the hydrogen is going to be positively charged. So yeah, so that's why. So when we're holding water molecules to one another, that positive charge on the hydrogen attracts to the negative charge on the oxygen. That's how they're associating. So hydrogen bond, think of it as hydrogen's always going to have a positive charge. It's never going to have a negative charge. So if it always has a positive charge and it has to associate with something without exchanging electrons, then it will be something that will be a negative charge based on just attraction there. But there's something else I was going to say, but I can't remember what it was. It's one of those days that I will get lost in babbling. All right, what element is in all organic molecules? Okay, so this is going to be carbon. That is right. And the only time you can call it organic doesn't mean that only carbon has to be there. You have to also have hydrogens in its backbone as well. So you'll have to have both, but carbon is the most important one there. All right, good job. All right, true or false? Water is a poor solvent. Last time I gave this school just a statement. I thought of a pun, just like poor, like P-O-U-R, like poor. Yeah, so it is an excellent solvent because of the polarity of it. And that's why it pretty much is the solvent of life. So that will be that. I keep forgetting I don't need to go over here. All right, what is water's pH? You know. Alrighty, so it's 7. So whenever we're having pH of 7 that is in the middle, because of it goes from 0 to 14, so right smack in the middle is neutral. That is 7. So that's exactly right. Good job with that. Okay, what range on the pH scale is an acid? Okay, yep, anything less than 7. So that's where the acids will be, 0 to 6.9, exactly. All right, lipids, what do we know about them? Awesome. So yeah, it's going to be all of the above. They make up our membrane. We know that. Phospholipid. So lipids already in there. We know that for sure. Used to make hormones. That's going to be the sterols, basically. Even cholesterol falls into that category. Those are lipids. And then can be used to make energy for the cell. Definitely we can break down fats to get energy. So you guys have it. All right. Which of the following organic molecules is amphipathic? All right, so most of you guys got this one. It's going to be phospholipid. This is where we potentially would have like that negative charged head and the lipid being non-polar, right? So it's going to be hydrophobic. So for amino acids, some of you guys chose that one. You're not completely wrong, but it would depend on the amino acid is really what we're talking about with that one. So yeah. So we can't put it all into that category. So what is a monomer of a nucleic acid? Okay, so it's a nucleotide. And remember, nucleotides are going to consist of a phosphate group with a sugar, it's pentose, so 5-carbon sugar. And that is also going to, so phosphate, sugar, phosphate, sugar, phosphate, sugar is what makes up the sides of the ladder of DNA. And then shooting off those sugars, we have the rungs of the ladder, the A, T, C, or G. Those are nitrogenous bases. That's what makes up the DNA structure. Okay. All right, last question. Alpha helices is what it should say. And beta pleated sheets are examples of which type of protein structure? Okay, so it's secondary. Again, this is just kind of one that we have to go over and be familiar with these terms. But if you can remember that alpha helices and beta sheets are going to be going together, those two things. And yeah, I might ask you a question about where does alpha helices go? I might not always bring up beta sheets, but if you can always remember that there would be alpha helices and beta sheets in the secondary, that there's 2 of them, then maybe that would help you remember it's secondary. I don't know if that will work for you, but I don't know. All right, so here's our little podium, so we can bask in the glory of our success for a little bit, and then we'll kick into talking about bacteria. I miss this music, that's the only thing. Maybe I'll have to like start it off on the computer and then like switch it or something. I don't know. This is really like depressing. Okay, so good job, everybody. Let's get into this. I'm gonna close this so it's not constantly playing music. PowerPoints. I'm gonna start over, and of course I've like zoomed in by 1,000. Okay. All right. So the whole reason that we have this up here, so I can like draw on things and zoom in on things. Hopefully it is something useful. We'll see. But you know, it doesn't hurt to try, right? So we've got chapter 4 talking about bacteria and archaea. But of course, before we even get into that, we need to remind ourselves what that even means overall. And yes, I have it on the board over there, but I'm going to go ahead and put it on here so that somebody's watching the recording later on, they can see it. So I'm going to switch to this. Okay, so basically what we have started out with when we learned in chapter 1, we had the microbes in general, right? And so if we have microbes, and that's fine, that's going to include guys that are made of cells and then guys that aren't. So the ones that are not are called acellular or non-cellular. That is an accurate term as well. And this is going to include 2 things, which we'll get into in chapter 7, viruses and prions or prions. I think I've said it before, but just in case I didn't, when you guys are pronouncing terms, because some of these words that you're going to learn in this class, or at least have to pronounce in this class, are ridiculous, like Escherichia coli or whatever else. So alkaligenes, whatever. And then so these those things, if you guys, as long as you're trying to pronounce them, you're good. Like as long as you know what you're talking about and what it relates to, that's the important thing. Okay, so that's really what I want to get across. But anyways, so we have that, and then we have the cellular guys. And the cellular guys, we can break into 2 categories as well. This will be the ones that have the nucleus. These are the eukaryotes. And then the other ones, I didn't write it properly, but hey, that's fine. We'll put the eukaryotes, which is more space. I can't go over that. So eukaryotes, okay? So that's the ones that we're going to be talking about today, because both bacteria and archaea fall into that category. Okay, so yeah, it's not pretty, but that's the breakdown of kind of what we have learned on how these guys are going to separate out. So this chapter 4, deals with bacteria and archaea. And let's not forget, when in chapter 1 we were talking about, you know, domain, kingdom, phylum, one that I need you guys to remember what to it, and that's going to be eukaryotes, bacteria, and archaea. And to put it into perspective, eukaryotes, in the environments they live in, why? This is what I'm saying is like, why can't it just be easy, you know? Gosh, just switch over, I guess. Let me plug this in, because I was like so happy to use this, and then why would it work? Just cut off in the middle. Off. Can't win. Okay, so let's So while we're getting into that, I'm just going to talk about some of the concepts that we need to know with these guys. Well, it's not actually connecting to my drive. It's plugged in. You guys see me plugging it in, right? Why does it have to be this way? If I didn't have to reload everything on this computer every time, this wouldn't be a problem. Because it doesn't save like anything. It's the only reason it has this even up here is because I used it this morning. And then every day it erases everything that I put on it. This is all of the professors, by the way, it's not just me. It's just I bitch about it more. Let me get this out of the way. Angle. Okay, that extra push. Let me share this on Zoom. What is happening? What's happening? I can't, sorry, I can't share it with people that might be online unless I close out of this one. So let me get out. And of course, you're going to learn that in the next chapter. But that's going to be things like mitochondria or endoplasmic reticulum. Anything that is a unit that is like defined in the cell, that is, membrane bound, has an exact function in the cell. That's what we're talking about with organelles. Bacteria don't have any at all. So keep that in mind. If they don't have any, they can't have a nucleus either. So that kind of goes back and forth how you want to look at it. Okay, so this is a good figure to come back to whenever you guys are studying, to kind of like at least give you the terms, and you can like block out the definitions and see if you can remember what each thing is or where it is basically in the cell and all of that. But we're of course going to be going through all of this nonsense. So bacterial cells are cells. And this information that we have here on this slide about what a cell is can apply to bacterial cell. This part here can apply to any cell. So if you are a cell, you have to be doing those things. This is how we're going to determine which falls into acellular and cellular in our chart where we separated all our microbes out. So if you are able to reproduce, and that means the cell can divide and make a new cell, and you have metabolism, meaning you can break down or build new components in the cell, and then nutrient processing, taking in nutrients from outside and using them to make new cells or whatever. If you can do those things, you're a cell. That's it. Viruses, no. Prions, absolutely not. Okay, and we'll talk about why in chapter 7. But yeah, so bacteria also can act as a group. They can exist in colonies or biofilms. We'll talk about what biofilms are in a minute. And we kind of have an idea of what a colony is, right? One cell grows up into a big pile. It's discrete unit from the same single cell, just multiplied. So they can do these things that they act as a group, but they are not true multicellular organisms. When you have a multicellular organism like you and I, if you take a cell off of us, it cannot grow into a new person, right? No. If you take a cell out of a colony, can it grow into a new colony? Absolutely. So these guys are not multicellular, they're just working as a group. So see it that way. They're always single cell. So are archaea. Both of these guys, any of the eukaryotes, all single cell organisms. It will never be multicellular. All right, so let's talk about some of the shapes that you can see these guys in. If you're looking at them in the microscope, I guess we're just going to jump right into that. The coccus and the bacillus are the two that you need to really concern yourself with, because you will be asked about them in lecture and in lab, and you'll be required to identify them in the lab for sure. I don't give, like, for the most part, I don't give pictures for the questions on the tests. So I just mostly concepts that I'm testing. But in the lab, you have to look at things and tell me what you're looking at. So that makes sense, of course. The two that I have in the lab to actually test you with are either going to be cocci or bacilli. Okay, so those are the two shapes. Curve is a thing that can exist. That's just talking about things that be spiral shape, a spirochete, a spirillum, vibrio, corkscrew, anything that has a curve to it. But we're not going to see that in our lab as much. So I really definitely want you guys to be familiar with the coccus and the bacillus. Cocci are round, sphere shaped, typically round is all I really want you guys to worry about with it. Don't get into other details. And then bacilli, that's a blocky spindle shaped, rod shaped. Okay. So the ones that look like long pills, those are bacilli. The cocci are the round ones. You can, I don't know if it helps you remember, if you look at cocci, the C's are round, the O is round, that's the round, it's a sphere. And the other one would have to be the rod shaped, because those are your only two choices really when you break it down. Is it round or is it rod? That's it. So the other thing that they could potentially be, and again, we won't see in the lab, but be aware of the fact that it could exist, pleomorphism, which means the size of the cell or the shape of the cell could just be random, sort of. It sort of depends on that cell's like makeup and genetic makeup and whatever it says there. So here's some pictures talking about cells as well as the arrangements, which we'll talk about what the arrangement terms are in a moment. But look at this, you see on this purple, why do we do it this way? We see on these purple guys, these round spheres basically that are all stuck together, kind of like grape, like bunches, right? And then underneath it, we also have some more round guys. So this is talking about this. Oh, sorry, I did not draw it that very well. But that whole section underneath being cocci, and then we have rods. These are examples of what rods would look like, and then the curved ones. So yeah, I mean, rods look like what you would expect bacteria to look like. Yeah. So then we have the cocci, they're little round guys, just so that we're aware of it. So yeah, you're probably hearing me say cocci or coccus, and you're like, yeah, like staphylococcus or streptococcus, exactly like that, exactly. So some of them have their shape and or arrangement in their name. So if your name was like lactococcus, then you would know that it is a cocci. It's going to be round already in its name. It doesn't necessarily talk about its arrangement there, but it does talk about its shape. But if we talk about something like Escherichia coli, that's not giving you any information about shape or its arrangement or anything. Only some of them will give up that information in the name, but it's helpful when they do. Okay, let's talk about the arrangements of the cocci. The cocci and the bacilli can arrange themselves in different ways than one another. So the cocci, they can do these things, these diplococci. So they would be, I could say they look like butts because that's what they look like to me when they do it. But they're real round and they can stick together almost at a line, a cleavage point. They do, they look like butts, just kind of all over the place. So those are diplo di, that's two. Next, the tetrads, it's just a group of four. Staphylococci and streptococci. This is one that might take you a little bit to remember what the difference is between them. That's okay, but you do need to know by the exam. So staphylo is going to be talking about those irregular bunches that we saw, either grape-like clusters or just irregular. Either one's accurate description of it, but it's like clustering together. Then streptococci are chains. And the way I remember is strepto sounds like a strip. And so a strip is like a chain. So that's how I know those are the ones that are going to be linked end to end. Like what we saw in the image over here on the bottom with the purple that are like in little chains. They look like little worms, but there's actually the round guys just stuck together end to end. That's Streptococcus pyogenes that is depicting. And so that's what causes strepto. And then we have Sarcina, which is like a cuboid. All right, then the bacilli are our rod-shaped guys again. These guys can also be diplo, of course, diplobacilli, and they would be end to end if they do that. So just be two guys, just like end to end, right? Then we have the streptobacilli. They can get in long chains like that. You notice we do not have staphylo for the bacilli. It just doesn't make sense with the shapes that they're in. And then we have palisades. That you can see that maybe can replace it in your mind. But if we have strepto where we're end to end, now imagine that we're linked together side to side. So it's like almost like looking like a fence. So palisade, that's what that means, fence, palisade. So yeah, so those are our arrangements for our cocci and our bacilli. I don't care about the arrangements for the other guys. We have biofilms that can form, and I mentioned it briefly just a minute ago, but let's talk about what they are. These are whenever microbes are going to grow together in an environment where there's a lot of food and protection, and they all work kind of together to signal one another how they're doing, so they can all know how their environment's doing. Biofilms are pretty important in medicine, whether any sort of living creature at all, honestly, and outside of that. But for medical purposes, when we talk about diseases, the number one most common infectious disease in human beings is dental caries, so tooth decay. And yeah, so that's going to be caused by biofilms of bacteria forming on your teeth. It can be involving several different species of bacteria, or other organisms can get involved, like archaea can get involved and stuff like that. It builds up on your teeth, and plaque along the gum line is what we're talking about here. And this is why it's so important that you floss, because you're not going to get rid of that biofilm by rinsing with a mouthwash or something. You have to actually get in between the teeth and scrape the biofilm away with the floss, and that's what flossing is for. Otherwise, the biofilm will just stay hanging out in between your teeth. Even at a small microscopic level, that can cause quite a lot of damage to your teeth and your gums. That's where gingivitis comes from. And if you are one of those people that's like, yeah, well, I don't like to floss because it causes me to bleed a lot and it hurts. Why do you think that is? Because that's my husband used to be. And I was like, yeah, I promise if you floss regularly for like a week or two, that will stop. Absolutely. Once you've gotten rid of those biofilms and you've got healthy gum tissue going on there, you'll be doing a lot better. So trust me, I bet you feel like you need to brush your teeth now. So when we're forming biofilms, it's pretty straightforward as far as how it's going to happen. You get some original bacteria that are going to come over to a surface like your teeth. Usually if it's porous or rough, something like that, it's easier for them to stick onto. So these bacteria have a sticky little coating they can make. And we'll talk about what that is. But that helps them stick to the surface. And once they're stuck, their little sticky coating can kind of spread and help them all stick together. And then other bacteria and other fungi and other archaea or whatever can all come and join in on the party. If you're having a big old party and everybody's happy and everybody's leaving their party waste around, like you're finding out like in this big old building that we're having this party and you're finding, evidence that there's a party and people still having a good time, whatever, beer cans or whatever you like at your parties, and you're like, yeah, everybody's still having a good time. Everybody's cheering, everybody's happy. What happens if people start, screaming and running out the building? There's evidence that things aren't going well. It could be not even that you're on the same side of the building where, screaming and running out is happening, but you notice that it's quiet. You'll notice that there isn't as much, of the trash that was happy time building up and whatever. And maybe in your corner, we start to realize that there's no more party and maybe you should kick it. You should not be here anymore. That's kind of how the bacteria will work. They will have waste produced from whenever they are having lots of food and energy and, safety and all of that. They have those kinds of waste. And then they would have, signals, chemicals that they make, or, if we're breaking bacteria actually apart from, let's say, brushing or flossing or something like that, that would tell the bacteria, hey, we're seeing bacteria parts down here. Maybe we should not be here. So then you're likely to be able to break up the biofilm a lot better when they start, dissociating from it. So that's the idea of how biofilms work. That kind of ability for them to communicate with each other, it's not true communication, right? But to read how the whole biofilm is doing, it's called quorum sensing. It's something we'll talk about later on, but I feel like it's a pretty cool concept. Let's talk about bacteria themselves. They have two different groups of appendages, okay? The ones that they use to move with, and the ones they use to attach or other things with. So only one of them really is used for moving, and everything else is everything else. Let's get into this. The flagella are the structures that bacteria use to move around. they move it with this little, it looks like a motor, sort of like a rotor there. And if you look at it actually in an electron microscope, it does look like that in real life. And what it allows you to do is that long filament that looks like a hair coming off of the bacteria, the protein-based filament has not got muscle in it or anything, so they can't actually move it around. So what they do is they just turn it, they spin it. So they can't whip it, but they can spin it, all right? So spin the whole thing, and then that is going to allow them to move. It can only spin clockwise or counterclockwise. And so based on what we're spinning like, we'll determine how we're going to move in our direction. First, let's talk about their arrangement around the cells. We have polar, and then we have peritrichous. So peritrichous, I know it's underneath that other one. It shouldn't have been when they ran the slides, but whatever. I didn't really look over it that well when I was looking at McGraw-Hill's slides. But peritrichous means it's all over. So that's not polar, so just FYI. So polar means at the poles, right? The ends. In case it's not clear by the fact that I'm going to talk to you about the ends of the cells, we are not talking about the round cells. Cocci do not have flagella. Only bacilli do. And not all of the bacilli do, but a lot of them do. Okay. So monotrichous, trich, like trick, like if you've ever heard of trichotillomania or something like that, it's hair. So they're hair-like, clearly. So mono, lopho means tufts. If you have a tuft at one end or a tuft at each end, then that is lophotrichous. We'll have tufts. Amphitrichous, we have flagella, one flagella at each end, so both ends. And then peritrichous, all over. So you can see in the image here, we have an A, very clearly a monotrichous, really long boy there. Then we have over here a peritrichous, very obviously all over it, flagella just everywhere. And that's probably the best way you're going to be. If you are supermodal and you need to be able to get around a lot, it is actually best for you to have it all over the place, because then you can actually start to control your direction a little bit more. If you've just got one flagella and you can just spin it one way or another way, how do you go left and right when you're trying to go left and right? You just have to kind of hope that you can. The more flagella you have, you can turn on some of your flagella and not others, and that can help you control your movement a little bit better. So that's kind of cool. Amphitrichous there in C, and I guess just more of them there in D. I guess that's supposed to be a picture of how they move, what it looks like. Chemotaxis and phototaxis. Right. So we weren't talking about the party, but now let's talk about more specifically, if we had in this classroom like a plate of freshly baked cookies, right? And I blindfolded all of you, and I said, hey, if you can smell your way to the cookies, you can have a cookie. That's basically what bacteria do, and that's chemotaxis. Because where does smell or scent come from? From chemicals, right, that are in the air. It's not just some, amorphous idea. It actually is true chemicals that are in the air that you can smell. Bacteria can respond to the chemicals around them and move towards or away from them. Like if I had cookies here versus dog poop. Would you want to move towards the dog poop? No. So then we're going to be going away from it. So positive chemotaxis, going towards it, negative, going away from it. So it could be like a toxin or something in their environment. All right, phototaxis. This is if you're mostly going to be helpful if you are photosynthetic. I mean, that makes sense. All right, so this is how they move, how they use their flagella to get around, to get towards something or away from something. They run and they tumble. So in running, we're just spinning our flagellum in one direction, and then you'll just go in one way. You'll just be shooting along, spinning your little propeller behind you, going. A tumble is when you reverse the direction suddenly of your flagellum. So you were going clockwise, now we're going to go counterclockwise. It causes the cell to kind of stop and kind of move a little bit. It can't control this super well. So they have to sort of use this in a way that to hope that they're going to be moving in the direction that they want to move. And so if they're trying to get away from something, they're going to have a whole lot more tumbles in there in an attempt to keep moving away from what they're trying to get away from. So this is an image showing you what a run and a tumble would look like. The reds are going to be tumbles, just when you're changing your direction. And then the run is in green. On the things below, on the bottom, those boxes, In figure A, we have a depiction where we're having a whole lot of tumbles and a whole lot of runs, but we're not really getting in any specific direction. That's literally because it's just milling around. It's not doing anything. It's just chilling. Like it's just hanging out. Okay. So it'll look random because it is. If you're trying to get towards something that you like, then you might do more runs. And then whenever you're starting to sense that there's less cookie smell, maybe put in a tumble to see if you'll turn closer and run. But if it's not getting stronger, maybe do it, put in another tumble. You know what I mean? So that's how they would do it, and hope that they're moving in the right direction. Very inefficient compared to what we imagine, you know, moving towards something would look like. But that's how bacteria do it. The flagellum is the only way for them to move. There's no other thing that allows bacteria to move. All right, so we're gonna have our first set of questions here. You guys know how it goes. Stand up, stand up. Let's shake out, I don't know, whatever was happening earlier with the computer. All right. Are bacteria always single cell? So single cell organism versus multicellular organism. Christy R. So yeah, always single cell. Good. That is Charlie. Next question we have, what is the term for round cell shape Angelica? Yes, that's exactly right. So that's delta. Cocci, remember, cocci and bacilli are really the only ones that are important there. Okay, what is the term when cells are arranged in irregular grape-like clusters? Okay, odalis? I don't know how to pronounce it. Okay, okay. Yeah, like the irregular bunches. Yeah, staphylo. So right, I would say like the new terms that we're kind of introducing as far as like the arrangements, right? We have strepto, staphylo, and palisades. The other ones are kind of self-explanatory. So if you can remember strepto versus staphylo, no, palisades on its own, then I feel like if you can tell the difference between those, you're doing pretty good. So when you guys get to studying on this stuff, that's kind of where I would focus my attention. So the next question, what is the term? Oh, we already did this, didn't change it. What appendage can bacteria use to move? Emily. Flagellum. Alrighty, good job. And then what does peritrichous arrangement mean? Taylor. All right. Fidel.
Does it mean all over?
Yeah, all over. That's exactly right. Okay. I believe that's the last one for that, right? There's, oh, there's one more. Of course there is. What is chemotaxis? Kayla. I have not gone through, we had quite a lot of like change up in people in the class, so forgive me for not, I updated it and now it's like completely wrong. Samantha. Nope. I believe that was one that I didn't take out from last time even. Okay, Dusty.
Chemotaxis is whenever the cell is trying to find it through smell.
Yeah, smell, chemical, as long as you can recognize it as that. You guys can have a seat down. Keep in mind too, when you guys are like going to be taking the test, keep in mind that it's multiple choice. And so you'll have words there that you can like look at and actually choose from. So it shouldn't be too bad as far as I'm not going to ask you, fill in the blank questions. All right, let's move on. So we have our attachment and channel formation appendages. These will be the pilus and the fimbriae. Let's get into them. The fimbriae is our attachment guy. It's just spiky fibers, protein fibers all over the surface that just allow it to attach to specific, usually specific types of surfaces and stuff. This is what E. coli uses to attach to your epithelial cells, particularly when we're talking about the epithelium inside the urinary tract. When you get urinary tract infections, that's what they're using as the fimbriae. 80% of urinary tract infections are caused by E. coli. I think I told you guys that. All right, so cool, that's for attachment. Okay, the pili, or the pilus for single, these can be used for attachment. I'm less concerned with that though. Definitely I would see it as flagella movement, fimbriae attachment, and the pili are for creating tunnels between or channels between cells. And then sharing genetic information through those channels. They can make copies of extra genes that they might have and then shuttle them through those channels to the other guy. That's how you might share things like antibiotic resistance, for example. So that is called the pilus, or if there's more than one, they are pili, and we call them sex pili often because they're involved in conjugation. And yeah, like similar to conjugal visits, we're sharing DNA. So yeah, it makes sense. So conjugation, we have the sex pilus, we have transfer of DNA through these channels. Crazy to think about that that's actually happening, but it is. Let's talk about some coatings that the bacteria can have on the outside. They can have an S layer, which is just some protein on the outside, and I'm not going to ask you about it. It's not on your study sheet. Next, we have the glycocalyx. This is the glycocalyx is a sugary, sticky coating on the outside, and there's two different versions that we can have of this: the slime layer or the capsule. Okay, so the sticky, sugary coating, it might have a little protein in it, but the thing that makes it sticky really is the sugary of it. Slime layer is loosely associated. It's more for like protection. It can be also for adhesion. We'll see it sometimes in biofilms. But the thing that concerns us most in what we're learning about microbiology for will be the capsule. The capsule is more tightly bound. It's denser than the slime layer. You can see a picture depicting that loose kind of association of the slime layer and the capsule really tight on the outside. Still a sticky coating. Still allows you to stick to surfaces. You can see here we have an image of what the capsule would look like on the outside of bacteria. That purple background is generated from a negative stain. You'll remember from that. So negative on the surface of your cell, negative stain, they repel each other, so we're staining the background. Okay. Then we have a positive stain that stains the cells, which are those purple lines in the middle of the white area. And then capsules themselves aren't going to stain. So they're white, they look like white around it. So all of that white is capsule. If you do not make a capsule, which some bacteria don't, but most of them do, to be honest, then you would just be cell straight up against that purple. Like it would be purple and slightly pink up against purple. There'd be no white at all. So that's what it would look like. And you can see in that plate at the bottom, it's showing us bacteria that don't have the capsule gene, and then if you give them the same bacteria a capsule gene, look at the difference. You can see that expressed clearly on the plate already. So yeah, these are sticky surface, allows you to attach to things. It allows you to make biofilms. So that's part of the deal with the biofilm, the sticky attaching. But probably most importantly for us, we have the phagocytic evasion. And what phagocyte or phagocytosis means, phago means to eat and cyto means cell. So this is whenever cells are going around eating things. So when are our cells going around eating things? Well, let's talk about our white blood cells specifically. That's probably the ones that do it the most. Our white blood cells are going around eating anything that they see as non-self or damaged cells, eating it up and digesting it down, right? That's phagocytosis. So literally come up to something like a bacteria and they envelop it with their whole entire body, their little feet, and take it inside of the cell and put it in a little vesicle and then break it down. Does that with bacteria and whatever else it finds out there that shouldn't be there. What types of white blood cells do this? Quite a few of them. Things like neutrophils or macrophages. We'll talk about them in chapter chapter 15 and 16. And then you'll also probably learn a little bit about them if you guys are taking physiology or when you guys take physiology, or if you did. Talk about neutrophils and stuff. Neutrophils are the most numerous of the white blood cells in your blood. If you get a CBC done, for example, it should be like 55% of your white blood cells is neutrophils just alone. And they are some of the ones that are like always century around. All right, so cool. So that's what phagocytosis is. If our white blood cells are going around eating things up, it's harder to grab the guy if he's got the sticky coating on him. So they can kind of just like slip out of the grabbing action and get away. That's why when I say, yeah, most of the bacteria are going to have the capsule. It's useful. All right, let's talk about the outside of the cell. We're going to get into basically our covering call it the cell envelope. The whole thing, everything that is on the outside of the cell, protecting the cell, controlling what gets in and out of it, the cell envelope, right? It can be composed of several parts. Any cell ever. If you fall into cellular on your microbes, then you have a cytoplasmic membrane. Every cell has a cytoplasmic membrane. And it sort of explains that to you in the word of it. We're talking cytoplasm, that's the contents, the goo in your cells, everything that you're doing is happening in, it's all suspended in your cytoplasm. Cyto being cell. And plasm, when you think of like plasma or something, liquidy. So the cytoplasmic membrane is the membrane that's holding all of that in. Obviously, we have that. That's all we have. We don't have cell walls or anything outside of that. So that's really important for us. So if you fall into cellular, you have that off the bat. Bacteria also have cell walls, and we kind of already talked into that a little bit, but we're going to get into the details of what they're made of and how they function and all of that. And then we have the outer membrane that only some bacteria have. Now, this is pretty key here. When I say some, I'm not talking about a few. I'm talking about the distinction between gram-positive and gram-negative bacteria. So I'm talking about a great divide. Depends on this. And yeah, of course, we're going to go over that. So the gram stain is the stain developed by this guy that allows you to see the difference. Basically, by color, if your organism is red or purple, whether you have a thick cell wall or a thin wall with an extra outer membrane. You can just tell that just by how it stains on here. So that's what the gram stain is for and what it does for us. And we're going to talk about what gram-positive and gram-negative really means. The word explanations are up here. Gram-positive means you have a thick cell wall of peptidoglycan in addition to your cytoplasmic membrane. And then the gram-negative, they have a thin cell wall of peptidoglycan with another outer membrane on the surface. There's the words for it, but we're going to show pictures. All right, this is a picture depicting them versus one another, if you will. This is on the left, our gram-positive organism. It's nice and purple for us because that's the color it would stain in the gram stain. We have all these crazy looking ball guys that are coming out of it in the orange or whatever. And down at the bottom, we have our cytoplasmic membrane. For both of them, they have that. So look at how thick that purple layer is. That's the cell wall. That's just the cell wall, the peptidoglycan. Imagine scaffolding or like lattice work, something like this, just on top of itself over and over and over again, like super high scaffolding. That's pretty much exactly what peptidoglycan is. It's that sort of thing. It gives a lot of structure and strength to the cell. I mean, you can imagine it would. Look at it, like a freaking, it's thick, right? So that structure allows for gram-positive bacteria to maintain the structure of their cells, even in adverse environments, like if I popped the cell into salt. So if I put you into salt, it's going to suck all the moisture out of your cells, and your cells are going to shrink up, right? They will eventually too with enough salt, but even in a slightly salty solution, let's say, they won't get shriveled up as much as quickly. So that helps them maintain their shape, and therefore they can survive better. Not all gram-positive bacteria love salt, but some of them, they really survive pretty well in it because of their features of their cell wall. And one of the examples would be staph. So Staphylococcus aureus, or the ones that's common on everybody's skin pretty much, is going to be Staphylococcus epidermidis. Those guys have, that thick cell wall, and they live on the surface of your skin where it's nice and salty. Gram-negative bacteria aren't going to be able to do that, because look at that. Look at their puny little, look at that. So you can see that the pink in the gram-negative guy, that little pink line through the middle, that's their wall. So much smaller, and this probably isn't even to scale. I mean, we're talking about like maybe one layer versus like 300 layers or something. So we are very, very significantly smaller cell wall here. We do have some, it is there, but it's nowhere near as functional, okay? So we have this outer membrane, like I told you, the gram-negatives would have an outer membrane. Why do they have it? Because where's their stability? Where's their structure? What's helping control what's going in and out of the cell? They don't have the cell wall doing that for them, so they have another membrane that kind of makes up for the slack, okay? Kind of makes sense? Some features that are defining features between these guys. You notice how the orange bead guys on the wall aren't there anywhere on the gram-negative. these are lipoteichoic and teichoic acid. These are important for maintaining the stability of these. Anytime that the bacteria are going to divide, which they do all the time, right? So anytime they're making a new wall, these guys kind of help out with that. And if you get damage to the cell wall, they help with rebuilding it as well. So that's what lipoteichoic and teichoic acid do. So gram negatives don't have that. So we've already found a feature, like even chemically, that you could test for gram positive versus negative. If I told you on the test, hey, your patient has a lipoteichoic and teichoic acid, what could it be? Gram positive bacteria, gram negative bacteria, fungi, virus. Gram positive, okay? So stuff like that is the stuff that you need to know for application for the test. Those differences. Because one feature that the gram negatives have, it's super tiny on this screen. We're going to talk about the makeup of it later, and I'm never going to quiz you about the chemical makeup of it, but definitely know what it is. Lipopolysaccharide, or LPS, or endotoxin. All of those are the same. So lipopolysaccharide, LPS, literally that's an abbreviation of that, or endotoxin. Think of it this way. Endotoxin is the term that the medical community came up to whenever they were dealing with it. And the lipopolysaccharide is the scientific community. Okay, so it's the same. It's just fancy words or not fancy words or whatever. But yeah, so let's get into that. Peptidoglycan, again, forms the cell wall. I will keep saying peptidoglycan and peptidoglycan. What does that mean? Peptidoglycan. We have peptide as one of the words, and then glycan as another part of the word. So peptide. A peptide is a few amino acids linked together, right? And we kind of learn about that. We learn about peptide bonds and what they're for, linking amino acids to make, proteins. But peptide is like a sequence of amino acids. So a little bit of protein there, that's our peptide. And glycan, if it sounds like it will be like glucose or something like that. Glycogen, anything sugar-based, it is. So these are sugars there. So sugars and protein, really all it is. And they are providing a lattice work that's pretty strong, but still flexible. This is what it kind of looks like. Again, I'm not going to make you draw it or anything, but I feel like it's kind of nice to understand what's going on here. So you can see the sugars are the little hexagon guys, the GM, because there's two different kinds of sugars that I won't test you on. These are N-acetylglucosamine and N-acetylmuramic acid. So they're just using the G and the M from that. They alternate and they make one long chain. And then the peptide is the green things along the side, linking those chains together. Like I said, it's a lot like scaffolding. That's the gist of how it works. So we've got that going. And we already talked about the teichoic and whatever. All right, gram negatives have a cell wall. It's usually a thin sheet, maybe 1 to 3 nanometers. Won't test you on that, but their bigger issue is, of course, going to be that they have that outer membrane. So there's some things that don't fall into this category with these guys. So you'll be gram positive or you'll be gram negative. That's the only thing you can state. But some of the gram positives aren't really quite gram positive, or some of the gram negatives aren't really truly gram negative. So here's our exceptions to the rule. All right, these guys will stain gram positive, but they're not true gram positives because they got weird stuff going on. They have something called mycolic acid on their surface. It's basically a lipid. It's like a waxy fatty acid. It helps with them being able to survive better, just makes them hardy and resistant and all this. All of these guys are Mycobacterium. And yes, also nocardia, but the only one you have to know for this course is Mycobacterium, because that's the medically significant one, not nocardia. So I'll only ever ask you about Mycobacterium. And myco has myco in it, just like mycolic acid does. But that's not a giveaway, because look at our next one, mycoplasmas. They do not have mycolic acid. So, so I'm going to walk you through how I remember which one's which. But mycolic acid, that wax, when we do the acid fast stain, that will stain with a lipid soluble stain that interacts with the wax that can't stain really well otherwise, and only will stain with the wax and not regular cells. So we use that lipid soluble stain. It will interact with the mycolic acid. And then when you rinse it with acid, it doesn't rinse away the stain if you have the wax. So that's why it's called acid fast, because it's going to hold fast to the color, even in the presence of the acid, which we'll do that in exercise number 4 in lab. So, so cool. So that's what that is. Why are these important? I say they're medically relevant. This is mycobacterium. This is, which we mentioned previously. This is tuberculosis and leprosy and stuff. It can also cause, Mycobacterium avium can cause tuberculosis as well, but it's not the true tuberculosis bacteria we're used to seeing. That really only happens if you have HIV. All right, so we have mycoplasmas. So yeah, I know they're myco, I'm aware, okay? These guys don't have a cell wall. So these guys stay negative, gram negative, because there's no cell wall at all. They are supported with sterols, just like our cells are. And so like we have cholesterol to keep our shape. It doesn't work very well for maintaining shape like the other bacterial cell walls do. So they are pleomorphic. So this is where we're coming back with that pleomorphic idea. You can have random shapes and sizes and stuff because there's no wall containing it. So this is how I remember the difference between mycoplasma and mycobacterium. You have 2 mycos that you're going to work with in the class, concept-wise. Mycobacterium and mycoplasmas. There's not going to be any others. Mycobacterium and mycoplasmas. To me, I can think of mycoplasma. I was looking at those 2 words and I can't remember which one is which. Mycoplasmas. If I gave you a cup of plasma, somebody's plasma that we drained out of their bodies, let's say, and you pour it out on a table, is it going to hold its shape? Oh, right, because this is liquid. And this is my point. That's I remember which one is which, because mycoplasmas, that plasma like you spill everywhere, it's not going to hold a shape. These guys don't hold shapes. They're pleomorphic. That's their characteristic. And that's because they don't have a cell wall. And that makes sense why they would be pleomorphic, no shape. That you can remember from there. The other one, mycobacterium, is the mycolic acid one. Okay. I don't know. That's how I go. That's how I do it. So I'm not going to go into like the makeup of this stuff. Archaea have weird stuff going on. That's not normal lipids that are found in any other cells. They have like hydrocarbons. We say unique branched hydrocarbons that are not like fatty acids. Well, aren't our fatty acids like carbons and hydrogens? Yeah, but these guys are like freaking like octane and stuff. They're not like our fatty acids. Okay? They are other kinds of hydrocarbons, like off in another realm. So when I say archaea are weird, man, I'm not kidding. We know the cytoplasmic membrane around any cell, probably you've already learned this, but it is selectively permeable. What does that mean? It means it's going to hold everything in that it should and keep everything out that it should. It will only let the passage of water and small nonpolar molecules. So that's going to come back when we learn about osmosis. We probably learned a little bit about osmosis, but we're going to kind of talk about that in relation to our cells here. Okay, this screen just turned off. That was sort of weird. It came back on, luckily. Yeah. Okay, so whatever. So that's kind of how we're containing everything in there and keeping everything controlled. Yes, the cell wall helps with that. And if you have another outer membrane, that helps even more. So that outer membrane of our gram negatives contains lipopolysaccharide, or LPS. And basically it works as a marker or receptor on the surface of their cells. But to us, we see this as a toxin. Our body responds to it toxically. And what do I mean by that? This is a very strong stimulant of fever and shock. When your body starts reacting to this, it's like it goes into overdrive reacting to it sometimes. And that can be deadly. Just seeing it around can be deadly. So that's endotoxin. It's not any small thing. All the gram negatives have this, and you gotta believe, man, when I tell you that gram negatives are friggin' everywhere. Pretty much all, not every single one, good large proportion of all the bacteria in your gut, most of them are gonna be gram negative. So they are all rocking endotoxin. All gram negatives have an endotoxin. So if you get an infection with any of those guys, whether it is E. coli or whatever, then you're looking at endotoxin being exposed in your system and potentially making you feel bad. Not always going to kill you with shock, but the idea is that it's a possibility. Okay, so cool. So we've started looking into why the structures of bacteria are important to us. So let's go back into the boring stuff. This is the gram stain. We're going to go through the steps and talk about how it works now that we understand what the wall looks like and how the envelope functions. Here, our first step is to add crystal violet. It is the color that it sounds like. It's purple. It's a purple stain. And you can use it just to stain if you wanted to. If you just put it on and leave it there, then you'll stain your cells purple. But if you go through these steps, you can go through the gram stain. All right, so we put our crystal violet on our cells. It'll stain the cells purple. Here we can see that in the gram positive, that thick cell wall, that peptidoglycan scaffolding is going to trap the crystal violet crystals or whatever in that scaffolding so that it can't get out. So it's trapped in there. In the gram negative, you can see that it's in there, but let me tell you, believe me when I say that it is very loosely in there. All right, so it might survive a water rinsing, but it's not going to survive the decoloring that we're going to do in a minute. Next, we have grams iodine. So both cells at this point are purple. We're going to add grams iodine. This is our mordant. This is the term to refer to what it's going to do. And I remember what it does. So it sticks things together. And what I remember is that you have a brick and mortar. So brick and mortar, the mortar holds the bricks together, right? So that's what the iodine is going to do. If you have been kind of saturated with crystal violet, it'll help hold it in place. But our gram negative, since it isn't truly meshed in there the way that it would be with the bigger guy, it's really not doing much for it. But either way, we've still got purple cells on both sides if we look at them in the microscope at that point. All right, our third step is our decolorizer step. That's alcohol, it's literally ethanol. We're going to just drop it down the slide, and it will cause the outer membrane of the gram negative to dissolve and all that loosely associated crystal violet to get rinsed out. So then we have the color removed from those, whereas the gram positives are going to retain the color. So now we're finally getting evidence of difference. So that color change, gram positive is going to hold on to that because we locked that color in place. It had the ability to do so because of the scaffolding of peptidoglycan. We don't have a scaffolding to hold it in for gram negative. So when we rinse away that outer membrane, the color goes away. So now they're uncolored. Gram negative are uncolored. And the gram positive is still purple. And let me tell you, when I tell you that is purple, when they are purple, y'all. There's no you will look at sometimes at the pink of the red gram negatives and be like, I don't know, is it kind of purple? And then I will even tell students sometimes, I'll be like, just wait till you see the other side. Then you'll know. Then you'll know. Just wait. But yes, so it's very purple. And then the other one's it's a red dye. It looks pink to us. We'll call it red because red is a negative color, I feel like. And that's how I remember which one's which. So positive purple, negative red. And that's how you would look at them and see the difference of color. When you stain with safranin, you're going to stain it regardless of whether or not it's stained purple. You're not going to look at it every single step of the way. You're just going to go through the steps. So this purple cell on the left, the gram positives, it doesn't stain red because there's literally just so much purple already there. It doesn't matter. You won't see it. Okay. All right. You're talking about the cytoplasm. It's mostly water. This is the bacterial chromosome. This is the, just like we have chromosomes, they have circular chromosome that is large and bunched in one area called the nucleoid region. One chromosome, circle. That's it for these guys. And it is just DNA. It's not a weird molecule. DNA in a big circle, and there's only one of them for all bacteria. Then plasmids. These are also little circles of DNA that can have extra genes in them, but they're not required for survival, but they can help survival. This is where we talk about antibiotic resistance or extra toxin production and something like that. Plasmids can often carry these genes, and yes, you can duplicate them and then shoot them through your little sex pilus to the other guy next to you and spread the information that way. So these are DNA. They are non-essential. If you duplicate your cell and you have one in you, then you'll duplicate it as well. So it is also, vertically transmitted. We use this a lot for genetic engineering. Ribosomes, they're in there. We don't really need to worry about them too much because we're going to talk about these in chapter 6, and I'm not going to test about it in this unit. Then we have endospores. These are structures of cells that help them survive hostile conditions. So it makes them hardy, it makes them resistant, and not all bacteria can do this. And actually only 2 genera can do it. And everybody in those genera can do it. So remember how I said mycobacterium can do mycolic acid, and they're acid fast. So they're the only ones that can do that, and all of them do that. When we're talking about endospores, we're talking about anything with the name bacillus, whatever, or the name clostridium, whatever. And there are quite a lot of illnesses associated with these guys. Pretty serious. So they can make endospores whenever there are harsh conditions, and no other kinds of bacteria can.
They go through 2 phases of life: vegetative, which is where I am right now, metabolically active and living life and having stressful, whatever, interactions. It's cool. Then we have the endospore forming issue. You ran out of food or you ran out of water, something like that. That's usually the trigger. It's usually food. They don't have any carbon or nitrogen. So then they start realizing they're running low and they can't keep reproducing. So they'll make an endospore instead. When they decide to do that, that process is called sporulation. They can resist heating, drying, freezing, radiation, and chemicals. Let's go through that one again. Heating, they can resist boiling, by the way. Drying, I mean complete desiccation. They can survive it. Y'all ever heard of anthrax? Endospores, okay? Freezing, literally don't care about it. Radiation, we're talking about actual, like, proper, not UV radiation, but, you know, cell killing kind of radiation. We worry about how it works right now. But yes. And then chemicals that normally can kill other things. If you have ever worked in a hospital, C. diff, right? It's everywhere. It gets everywhere. And you have to go through all these special precautions to keep the spread. If you have a patient that has C. diff, keep it from spreading from one patient to another. And it's just a pain, or infection control to keep it under control. And if you have worked in a hospital, then you know exactly what I'm talking about. This is just a serious issue in hospitals, maintaining, you know, sterility and preventing spread of C. diff, because C. diff is Clostridium difficile, and it is able to make endospores, and it's hard to kill them even with the cleaners that they use in hospitals. All right. Yeah, like we said, nutrients. And then let's say that I am like starving to death and I want to make myself a little endospore. You make it within yourself, typically. It's like the structure within the cell. And that would make me a sporangium if I'm doing that. So the little baby endospore that you're building is the sporangium. The little baby endospore that you're building is the endospore, and you're the sporangium piece. It takes about 6 to 8 hours to make an endospore once you start going through it. And this is the process you'll go through: from normal cell, duplicating your DNA, and then walling it off, and then putting special coating around it, and then it'll get released out of the cell and wait until it gets waked up. Basically how it works. So germination is whenever you reach favorable conditions again. Yeah, so if we're talking about anthrax, for example, if you have powder in the envelope and it gets into Congress like it did, and you know, people are breathing it in, once it gets into like your lungs or anything, now it's got favorable conditions. It's got you know, all the food that it needs, and it's got, you know, water basically available to it, and that'll wake it up and it'll start to make bacteria. How long do you have until those endospores turn into an actual bacterial cell? About an hour and a half. So if you step on a nail out in your grandpa's backyard and it goes pretty deep down into your wound, well, the Clostridium bacteria associated with the disease that you would be getting, it requires that you can't be in oxygen. So it has to be a pretty deep cut. So if you just cut yourself, you're probably fine. But if it's pretty deep, you might want to be sure that you have your tetanus vaccine, because it's a very serious illness and you don't want to mess around with it. I know we don't really take it for granted because everybody's so vaccinated and all of this, but if you get tetanus, it's not just lockjaw that you will get. Lockjaw is like where it starts. And if they can't contain it, and it's easily uncontainable if you wait too long. It is a spastic paralysis that will form, which is like your muscles get stuck. So they just don't ever relax, and you will die of suffocation because your diaphragm will die. There's another one related to that, says Clostridium tetani, obviously. There's another one related to it. That bacteria is called Clostridium botulinum, and botulism causes flaccid paralysis. It's kind of like the opposite side of the coin, but you'll die the same way. So it's flaccid though, not the spastic. You'll still suffocate. These guys are terrible, is what I'm saying. So no Clostridium and no bacillus, okay? So that's all they're looking for, is the thing that's going to wake them up: water and or nutrient. We already talked through some of these. One of them I didn't just bring up would be C. perfringens. Clostridium perfringens causes gas gangrene. It's also a cause of food poisoning. And these things are like all over the place. I don't really want to go over I know you guys don't want to do the questions, but let's not do the questions just because then we just go to lab, because it's so late already. It's almost class over. So let's just skip them. I'm just going to kind of go through them. How about that? So you can get something from them. What is the difference between a gram positive and a gram negative bacterial cell? Biggest difference: thick cell wall on the gram positive, gram negative, thin with the outer membrane. That's the gist. What is the function of a bacterial capsule? That's that sticky coating. It's for adhesion to surfaces, like making biofilms as well as evading phagocytosis or white blood cells eating them, evading the immune system. Why do mycoplasmas have a pleomorphic shape? Because they do not have a cell wall to help them. And what is an endospore? It's that hardy, dormant structure that some bacteria, Clostridium bacillus, will make in harsh conditions, so they can wait out the harsh conditions and get woken up later on. So these dormant structures, I want to be completely clear. When you make an endospore, it's literally dormant. It doesn't do anything until it gets woken up again. So it's not motile. It's not doing anything. It's just chilling. It's like if you go into hibernation, right? All right. Oh yeah, there's this crap. I forgot there was more after it. There's the, I forgot about this. Every time, I'm not kidding. I've given this lecture 4 times already this semester, and I always forget. Archaea, they have weird components. That's what this is talking about, and they live in weird places, like legit. They make methane. Bacteria can also make methane. If you've ever had methane come out of a cow's butt all the time, it's the bacteria in their gut that's making it, not them, right? All right, extreme halophiles require salt to grow. So I'm saying they are halophiles. They love to grow in salt, but these are extreme halophiles. These are guys that like grow in like salty lakes and stuff like that. Like when we're talking about true salty lakes, they are good. They can't live anywhere near where we can survive. Normal like watery environments, they would die in because they've adapted to the salt. Psychrophiles grow in freezing temperatures. Hyperthermophiles, thermophiles like high temperatures. Hyperthermophiles, they will grow, they're flourishing between 80 degrees Celsius and 113 degrees Celsius. Flourishing above boiling temperatures, flourishing there. That's nuts. So when we're classifying our bacteria and our archaea, we can look at these guys at their cell envelope, which is their outer covering, and basically classify them based on that. There's these 4 terms. They all are cutes. Okay, that's the end of them, always cutes. Let's go through them. This is how I remember. All right, Mendoza cutes. This is not a joke. I actually did have a boyfriend when I was like 12 years old. His name was Joey Mendoza. Isn't that cute? It was a long time ago. It says archaic to me. That's archaic means old. So archaea. So I say Mendoza cutes. That's the one I always go to. If I can't remember which one is which, I start there. Mendoza cute. That is archaea because of that story. Next, firmicutes. Firm. Which ones have the firmest membrane outer envelope? That's going to be the gram positives with that thick cell wall, right? So they have the firmest, so they fall into firmicutes. None of the others qualify. Next, I go to the G that's left. The G you would think is gram related, and you would be correct. So it's not going to be gram positive because that went into firmicutes. So it's going to be gram negative. And then I sort the remainder into tenericutes. These are ones that lack a cell wall. So here we're talking about like mycoplasma and some others we're not going to learn about. And tener apparently has something to do with tender. I don't really understand about the words, but I did read that somewhere. So tender, soft, that might help you as well. And then we have Berge's manual that's going to talk about basically separating bacteria by their traits. So you're going to get all this info about your bacteria, and then where would you go to actually figure out what your answers are? Literally, like where would you go? It would be Berge's manual. That's all you need to know about it. I'm not going to ask you about that. And then there's subspecies and strains. These are like they qualify as E. coli, but they have an extra feature or something, and that's how they would. E. coli 0157H7 is the dysentery causing E. coli, bloody diarrhea. It's the serious one. When we're talking about food poisoning, people dying from it, that's E. coli 0157H7, STEC. But there's other E. coli, there's like normal E. coli that's normally kind of like feces and stuff that shouldn't be any more dangerous than like a urinary tract infection or something. They're different, yeah. So it's the strains, like whether they make toxins or not. So that's all that is. Oh, that's it. Okay. So yeah, this last chapter question was one I left on from others. But what's the difference between a chromosome and a plasmid? Remember, the chromosome is big. It contains the genetic information that the organism needs in order to live. It doesn't need the plasmid. It's just helpful. I'll see if I can't figure out what other better thing I can do with the iPad for next time. I'm not going to figure it out till after freaking break. everybody be sure that you sign the sign-in sheet so I can get you credit for it. And then we're gonna go up to the lab in case you didn't know. And you do need lab coat and closed-toed shoes. If you have them. You're fine.